What I really want to demonstrate today is how we are utilizing Nanopore whole genome sequencing at Lonza to enhance our cell line engineering. And after this legal disclaimer, I would like to start with a small intro to Lonza Biologics. So Lonza Biologics is a CDMO which produces high-quality therapeutic molecules, such as monoclonal antibodies, for our customers. We do this using the gold standard production system, which are our proprietary Chinese hamster ovary cells, in brief, CHO cell lines, that we grow up in large bioreactors where they would be producing the therapeutic molecules at a large amount. One of the first steps in this pipeline is the construction of biologics vectors. We usually utilize transposon vectors, which carry the genes of interest encoding the therapeutic molecule. In this example, it's a heavy chain and light chain gene of a monoclonal antibody. We take the vectors together with the mRNA encoding for the transposase enzyme and transfect our host cell line. And after the transfection, we select the top performing clones. We then rigorously characterize these clones and quite often observe that they are quite variable. This box plot on the right here gives you a taste on the variability in the amount of product produced by different clones. And our goal in R&D really is to link this phenotypic variation with the multiomic variability to improve especially the strategies for cell line and vector engineering, but also to come up with predictive models of clone performance. So for this, we recently onboarded Nanopore whole genome sequencing, and that actually allowed us to gain quite some unprecedented insights into our CHO-based production system. So fundamental to every omics and genetics work is a good reference genome. So one of our first endeavors in using Nanopore sequencing was we took 2 of our cell lines, we performed whole genome sequencing with Nanopore and created de novo assemblies of the CHO-K1SV and the CHO-K1SV GS knockout, 2 of our proprietary cell lines. We found that both assemblies had a very high completeness, so in the left plot here, comparable to the human reference genome and to the best public CHO reference. And when we use these internal references, we find that they enable a more accurate analysis because more of our omics reads map to the internal references compared to the public CHO reference. So by this, we could improve our omics analysis, and now having built these reference genomes, we can use those together with Nanopore sequencing to improve our method development in R&D. One of the methods we are trying to optimize is the transposon-mediated transfection technologies. Here, as I mentioned before, we have these vectors that have the genes of interest which we want to bring into our CHO cells. The inserts, which go into the, basically integrate into the CHO genome, are roughly 10 kb large. And to optimize this method, we want to know how often do those integrations happen and where in the genome, for example. So here the long reads from Nanopore sequencing are particularly useful because we can sequence, when we sequence a CHO cell line, we can sequence the insert together with flanking regions of the host genome. By this, we can precisely say, as shown in the plot in the middle, we can precisely find out where in the genome the integrations happen and how many of them happened. But we can take that even a step further and look at the DNA methylation readouts that we also obtain from Nanopore whole genome sequencing, and ask, do all of those insertions contribute equally to the, in this case, antibody titer? And what we find is, in this example, that the very right plot, that the green and the blue insertion, I'm not sure if you can see the colors, but it's the lines that are a bit higher. For the two of the insertions have a higher methylation. So what that means is they may be less expressed than the other three insertions. So what does it mean for method development? It means that when we are developing this technology, we not only need to care about the number of insertions and the genomic loci where they integrate, but also we need to optimize the DNA methylation patterns. And we are also using nanopore sequencing for identifying, so implementing in our multiomics analysis and identifying targets for cell line engineering. Here, especially the DNA methylation data are useful, which we started to integrate in our multiomics target identification pipelines. I want to give you a visual example of one of the experiments that we looked at, which is sequencing of two cell lines with different sequencing technologies. One cell line, the GSKO, has a knockout of the glutamine synthetase gene, and the second cell line we sequenced is its parent, which has a functional copy of the gene. We first performed a short-read RNA-seq, is the bar chart on the left, and find that the knockout version of the gene actually is not expressed anymore. So now we can ask the question, why is that the case? And here, when we now incorporate the DNA methylation data from nanopore sequencing, as shown in the plot in the middle, we find an explanation because the knocked out gene version has a much higher DNA methylation compared to the GS gene in the parental cell line with the functional copy. We can, of course, add further omics technologies to this, which add some additional explanation. One method that we recently started to explore is a short-read promoter capture high-C. That's the plot on the right. What this is doing is a high-C approach, so 3D genome approach, but measuring 3D interactions of promoters only. So what we find is that when we knocked out the gene, there's also reduced 3D promoter interactions in the region of this glutamine synthetase gene. Is denoted by the loss of the black lines there in the plot. And yeah, I think this example is just for one single gene, but it really nicely demonstrates how the different technologies integrate with each other. Of course, we are now, what we're doing is we're expanding this to all of the genes in the genome, not just to one example here. And we define targets if they have, for instance, consistent biological patterns like this one across the whole genome. And with that, I would like to conclude that nanopore genome sequencing for us at Lonza Biologics actually enabled a very precise assessment of the genetic quality of our CHO clones and vectors. Through integration, especially of the DNA methylation data, we can also come up with better engineering strategies and better targets. And all of this means that we can streamline the development strategies, which leads to reduced timelines and further downstream also cost savings. With that, I would like to thank everyone who contributed to this work, and thank you for listening.